It's my distinct pleasure today to introduce Professor Ruth Baker. Ruth has a master's in mathematics from the Mathematical Institute at the University of Oxford, where she stayed to do a DPhil, and where she stayed to become eventually a professor in applied mathematics at the Mathematical Institute at the University of Oxford. She has a long distinguished CV for someone her age. She's obviously very accomplished, many grants in applying mathematical modeling and using real qualitative data and quantitative data in order to sort of drive those mathematical models. Lots of papers in journals like Nature and Science, 170 manuscripts in all kinds of journals at age index of 32. As of March last year, she is a Royal Society Wolfson Merit Award holder. She had a Leverhulme Research Fellowship, and she was awarded in 2014 the Whitehead Prize for the London Mathematical Society. So obviously a very impressive CV. And although normally our seminars are on mathematical oncology, today is a more broad mathematical biology seminar. And I'm definitely looking forward to see the presentation, Ruth.
Thank you. Thanks for the introduction. I feel like there's a lot to live up to. But no, seriously, thanks, David, for the invitation and for the introduction. So basically, this is a bit of a talk of two halves, where the idea really is to showcase some of the work that we've done quite recently that really aims to sort of calibrate models to data quite carefully and to sort of to gain new insights from doing that. And really the emphasis is on maybe building a simpler model, but through the calibration process being able to learn something about the biological system. Ooh. Okay, hopefully this works. So, but yeah, as David said, I'm not sort of from the cancer field, which I guess is presumably where many of your speakers come from. So I just sort of introduce the focus of my research group. So we're interested in understanding how sort of groups of cells undergo collective cell motility and how those processes combine with adhesion, proliferation, and cell death to give rise to very complicated biological processes. And we mostly focus on aspects of embryo development, but then more recently have started thinking about aspects related to sort of tissue regeneration, such as wound healing, or even across to aspects of sort of cancer biology. And as sort of the field has begun to change, so since I started my PhD, where in developmental biology models were very much qualitative and the data was also essentially often qualitative, things have changed quite a lot, and now we are really seeing an explosion in quantitative data, and our ability to sort of characterize systems with a finer level of detail is really increasing. So our focus has become more and more thinking about how to make use of the sort of different types of quantitative data that are generated now or can be collected using sort of models that are calibrated to data and hopefully relatively realistic. But on the other hand, I guess what this talk is going to illustrate is that these models can still be quite simple at the same time. I think I'm sort of preaching to the converted, but we really try and think the applied projects about taking very much an interdisciplinary approach, where we're collaborating with different experimental groups who are looking in sort of both wild type and perturbed scenarios, various or a particular question. And then our involvement is very much developing the models, developing the means to simulate these computationally, doing the analysis of those models, and then working very closely to think about how to do data analysis and model testing with our collaborators. And for me, this is really integral in this cycle where you might have some experimentally based hypotheses that then you encode into a mathematical model. You test those hypotheses within the framework of that model, and you normally learn the most when you get something wrong. And this talk's going to illustrate that, I think. But that leads you to refine your ideas, refine your hypotheses, and then you can make essentially predictions using the model that can be tested experimentally. So hopefully I'm going to sort of show a little bit of that as well today. Okay, so it talks about the fact that we're kind of increasingly seeing sort of quantitative data that's collected, and whether that's in developmental biology or in sort of cancer biology or in tissue engineering, I think it's in the case sort of throughout. And I guess part of what I've sort of come to realize is that often to be able to kind of make the most use of that data, you want to be able to estimate the model parameters from the data. And calibrate your model to the data. So then you can essentially make predictions that are quantitative and can be tested on that level. And then that sort of leads to asking questions surrounding, whether your model's at the right level for the data. And that's long been a huge skill, I think, of applied mathematicians and mathematical models more generally, is thinking about, like, how do I pitch the model given the kind of understanding in the data that I have. And that was certainly the case when we had qualitative data. But also maybe now we have quantitative data there, I think there are maybe different ways to think about sort of answering that question. I'm going to illustrate that a little bit. But when I talk about my model being at the right level for my data, I'm often asking, can I pin down the parameters of the model given the data, or is there going to be uncertainty in those parameter estimates? And if there is uncertainty, which generally there is, how do I quantify, or can I quantify the uncertainty in those estimates? And then the important thing, I think, is to think then, okay, given my estimates are uncertain, how does uncertainty in parameter estimates feed forward into uncertainty in model predictions, right? Because that's where we're really asking questions about how much we rely on the model predictions in sort of validating the model or our hypotheses against data. So sort of thinking about some of these questions is going to be the focus of the talk today. There you go. And then in very simple models. So you have to excuse the fact that there's not much maths in all of this, because the models are relatively simple. So often when we're thinking about calibrating models to data, we're thinking about the inverse problem. So if I have a data set, can I estimate the model parameters and then use the calibrated model to make some predictions? So this is just one example, and I'll come back to this in the second half of the talk, where we might use a scratch or a wound healing assay, where typically you grow a population of cells to confluence in the dish. You take a pipette to remove a portion, or literally scratch or scrape away a portion of those cells, and then you watch essentially as the population of cells proliferates and moves to fill the available space, so to heal the wound. And what's quite nice about these kind of scratch assays from a modeling perspective, I guess, is that this is just and I'll show you later, this is just a sort of snapshot of the middle of the dish. But what you can do is often average the data in the y direction to get these nice sort of one-dimensional density profiles to work with. And then we're interested in inference in a Bayesian framework, where we want to estimate the posterior distribution. So this is essentially the probability of model parameters theta given data d. And we access that because through Bayes' rule, by writing this as proportional to the product of the likelihood and the prior. So this is the likelihood of the data given model parameters theta. And then the prior distribution just encodes any existing knowledge we might have about sort of parameter values in the model. So if we're thinking about this motile cell population, we might be using a PDE model, and then we've got a diffusion coefficient that tells us something about how quickly the cells move. And we know that that's got to be non-negative, but we might also be able to put an upper bound on that just because of sort of the limits in as to how quickly cells can move, for example. And so all of that sort of prior knowledge gets encoded in this prior distribution. So we're basically always working in this kind of framework. Okay, so what I'm going to do now is spend a little bit of time talking about a project that we worked on over the last sort of 3 years or so, maybe slightly more. And this is a project that focuses actually kind of on a smaller spatial scale than I alluded to. And this is thinking about within the cell, how do cells essentially localize mRNAs to the right place and do that in a robust fashion. So first of all, you might ask the question, like, well, why do cells want to do that? So actually many cell types use this process of putting mRNAs in the right place at the right time in a cell to essentially, not because they want the mRNAs there, but they want the proteins that they translate in the right place at the right time. And in a particular, this is thought to be a sort of more efficient process than sort of translating the proteins and then moving them, I guess, due to the stability and the fact that if you've got relatively large cells, you might have to transport many protein as molecules over large distances, whereas you tend to have fewer mRNA molecules. So there are many cell types. Things looking at even cells when they migrate, for example, but also looking at sort of synaptic plasticity. There are quite a few examples really of different cell types that use this sort of process to get proteins in the right place at the right time. And so we were interested in asking questions about what's controlling that localization process and how does a cell ensure that it's relatively robust so that it can polarize in the right way or move in the right direction or set up the body axis sort of correctly. And we sort of came upon this question really because there's an experimental group in Oxford of Elan Davis over in biochemistry who's really interested in these sort of questions in the context of Drosophila. So this is Drosophila sort of egg production line, or a Drosophila production line, if you like. So what you've got is a load of strings of kind of sort of Drosophila follicles sort of developing, and down, so at the top there, there's sort of younger ones, and as you move down a given string, you can see that the kind of age of each just sort of potential Drosophila, if you like, to keep it simple, is sort of increasing. So you have about 18, I think, of these strings in one of these sort of groups here, and this is like I said, the sort of the production line for generating new Drosophila. And we were interested in answering this question in Drosophila basically because it's relatively easily to access experimentally, and we can look at lots of sort of genetic perturbations in the context of Drosophila. Okay, so this is a sort of production, a picture of one of these production lines on its own. And again, as you go sort of from left to right along this string, you can see just the Drosophila sort of egg chamber is sort of, you can see how it's developing over time, if you like. And you can see the sort of structure getting increasingly complicated with different sort of cell types and different sort of cell rearrangements and sizes over that period. Okay, so what's really interesting about the Drosophila egg chamber? Well, if you look at it in this sort of schematic, so this would be around the sort of 16 cell stage. Of those 16 cells, actually only one of them is going to actually make up Drosophila, the organism, and the other 15 are essentially just support cells, and we call them nurse cells, and they're just helping the process along. And then within the oocyte, which is the developing Drosophila, you've got the nucleus over here. What you see is that there are a number of mRNAs that are spatially localized, and they're actually the mRNAs that set up the various body axes. So bicoid is well characterized in that sense. This is this one down here. So what we know is that Drosophila very successfully uses this process of mRNA localization in order to set up its body axes. And we wanted to understand, or use Drosophila to understand how this process is governed, if you like, and what makes it incredibly robust. But it's not just such as sort of simple as just looking at what happens in one cell, because actually as the sort of Drosophila egg chamber sort of develops from one cell to 16 cells, it does so by a series of cell divisions, but all of those cell divisions are incomplete. And what you get is these sort of intercellular bridges, which are known as ring canals, which essentially connect the 16 cells together. And they allow the passage or the transport of various sort of molecules and cargoes. And in particular, one of those is these mRNAs. Okay. And what was I going to say? And yes, and actually sort of what happens in Drosophila in the egg chamber is that these maternal or these mRNAs are actually produced in these 15 nurse cells, and they're transported through these intercellular bridges into the oocytes. So in this diagram, can you see my mouse? Actually, I should have asked that before. Let's see.
Yes, we can see it.
Yeah, you can. Okay, fab. So excellent. So in here, this is actually the cell that's going to be the oocyte itself, and you can see that even if an mRNA molecule is produced down here in the bottom corner, it will eventually be transported into the oocyte itself. So it's the nurse cells that are responsible for making these mRNAs, and then they're transported through this network into the oocyte and put in the right place. So this is a picture of what one actually looks like in reality. So we've got here, I'm going to focus just on one mRNA, which is known as gherkin, down here, and you can see the red dots are essentially sort of showing you where the mRNA are. And you can see there's plenty of red dots of gherkin mRNA in these nurse cells, which are these 3 that you can sort of see in this picture. And there's lots and lots and lots of gherkin already accumulated in the oocyte at this particular stage. And this is what a ring canal looks like as well. So these are these intercellular bridges that connect this parcel of 16 cells together. And hopefully you'll be able to see this movie, but this is just a Z-stack going through one of these egg chambers, and you can see these are the ring canals really, really clearly. And you can see the sort of 16 nurse cells. So what you can do is collect really, really good data that just tells you how the distribution of mRNA looks across the nurse cells and the oocytes over time. And you get, it is possible, because my PhD student can do this, to be able to identify each one of these nurse cells. And so you know which, when you're counting mRNAs, which nurse cells you're actually looking at. Okay. So I think I've said much of this before, but essentially during this process of Drosophila sort of development or agenesis, then the mRNAs, and we're going to focus on gherkin, are produced in these maternal nurse cells, and then they're transported through this this network of ring canals and into the oocyte. And we want to know sort of what drives this process, or whether there's one particular part of this process that is kind of rate limiting. And how and why it's robust. Okay, so and I'll try and remember to come back to this again, but we thought about models on a huge number of different levels for this different process, and we had incredibly complicated models that looked at kind of individual nurse cells and the transport process within an individual nurse cell, which is very, very complicated. So in this context, you have multiple mRNAs which are essentially generated in the nucleus, exported through nuclear pore complexes, and assembled into cargos and transported on the microtubule network of the cell. So you've got this, you know, these hugely complicated processes, and we thought, okay, let's build a model of, you know, each one of these nurse cells and the oocyte, and let's stitch it all together, and rapidly realized that was incredibly complicated computationally, and just generally maybe going to generate an unwieldy model. So Jonathan, my student, actually we really went back to scratch on this one, and back to the drawing board, and said, you know, I think what we should do is build the sort of simplest possible model that we can. So instead of having anything that was particularly well spatially resolved, we went for a compartment-based model where we're simply going to track the number of mRNA complexes in each of these 16 cells. So this is the wiring diagram that you can sort of vaguely keep in mind, where we've got 16 cells. The one labeled number 1 in red is going to be the oocyte, so the actual Drosophila sort of organism in the end, and then the other 15 in black are labeled 2 to 16 are going to be the supporting nurse cells. So all we're going to do is essentially count the number of mRNA complexes in each of these cells, and we're going to allow sort of transports between cells if they're connected by one of these intercellular bridges, which we call ring canals. And then we're going to produce the RNAs in each of the nurse cells. So if you write down a model for this, it just looks like a system of 16 ODEs, which can be really sort of succinctly written as on the slide. So Y just is a 16 dimensional vector, which is just counting the number of mRNA complexes. And then the change over time is just governed by production at rate A and transport at rate B. And then this vector V just encodes the fact that you're only producing these mRNA complexes within the nurse cells, so in cells 2 to 16. And then you've got this matrix B as well that encodes the sort of transport process. And the sort of actual sort of form of this doesn't matter too much, but really the idea is that B encodes 2 things. Number 1, you can only have transport between cells if they're connected by one of these ring canals. So that gives you kind of where the non-zero entries of B can be. And the other thing that we wanted to take into account was the fact that people believed that this transport process was biased. And then sort of in a way, it needs to be biased to ensure that you end up with mRNAs accumulating, mRNA complexes accumulating within the oocyte, so cell number 1. So nu represents that that bias parameter, but sort of counterintuitively in the kind of experimental field, nu would be biased away from the oocyte. So if you see low numbers for nu, that means bias sort of towards the oocyte.
You're probably going to mention this, Ruth, but yeah. The way you've written this implies that the production and the transport's actually uniform for every one of those.
Yeah.
Is that true?
It was really just a first approximation. So I guess you're assuming that you've got one copy of the gene in every cell, and that on average it's uniform. Clearly it's probably, you know, that's just definitely a first approximation and an oversimplification, but I guess we wanted to see what we could do with a model that looked like this. And then if you have to go back to characterize that further, then you can, but if you can capture kind of gross behaviors like this, then I think that's good. But yeah, certainly a good question. And I'll come back to that actually in a bit, because we did think about, certainly with production, we did think about that quite a bit more. So the nice thing about having a model like this is it's, you know, you can just write down the solution to it in one line, more or less, which is fab. And then you can start to think about how to connect the model to data. So there's the solution, in fact. And I guess just to show you before, you can also sort of look at sort of typical behaviors of the solution. So in here, I'm just kind of always have the cell ID on the x-axis. And then the only thing really to remember is that cell 1 is the oocyte, and that's where we're getting accumulation. So it all looks relatively sensible as long as you choose the parameters relatively sensibly. Okay, so I said that we wanted to do Bayesian inference. So Jonathan spent quite a bit of time, my student, collecting data from these systems. So really looking at counting the number, going through multiple egg chambers and counting the number of mRNA complexes that were in there to get quantitative data. And then we wanted to think about how to infer the parameters of the model given these data. So again, we're using this Bayesian framework. And so in order to make progress, we had to impose a measurement model. So essentially, how many of these mRNAs are you actually sort of measuring when you image and you collect data from one of these systems? So we assumed it was negative binomial. So there's introduces an additional sort of variance parameter sigma, but also we had another parameter in there, phi. And it's sort of the devil in the details with this one, really. But what happens is that so these mRNA cargoes, they're essentially, mRNAs are produced in the nerve cells, they're packaged up with some other proteins, and they're transported on the microtubule network of the cells, between the cells, across these bridges. And what happens somehow is that as they go from cells 5, 9, 2, and 3, so the ones that are next to the oocyte, and as they're transported into the oocyte, they're almost like there's a higher order assembly process where multiple of these cargoes are sort of packaged together. So whenever we see a sort of bright spot in the oocyte, that there's more mRNAs in one of those bright spots than there is in one of any of the other nerve cells. So the measurement model actually really needed to take account of that, and there it's represented by this parameter phi down here. So now we've got more parameters. So we had production at rate A, transport at rate B, we had bias that was new, and then we've got sigma and phi, which come from the measurement model. So we've got 5 parameters to infer in this framework, and we wanted to see how we could do. Now I'm going to come back to this before, but there was then a problem, right? Because there were some identifiability issues. So essentially, if you didn't have an independent measurement of this sort of higher order assembly process, then you couldn't sort of confidently estimate sort of all of the other parameters independent of one another, if you like. So the first thing we set out to do was to sort of just use existing data to try and estimate that parameter quite well, kind of beforehand. And the idea really here is that if you look in one of the nerve cells and you look in one of the oocyte and you look at the background intensity of gherkin, so now in purple, what you're trying to do is look at the intensity of those spots that you see and try and from there get a good estimate of essentially how many mRNAs have you got in each one of these bright spots in the oocyte compared to the nurse cells. And it was really important to do that and to characterize that parameter quite well in order to be able to estimate the rest of the parameters of the model confidently. So this is just a plot to show these results. And what it turns out is that there's on average about 2.5 of these sort of little cargoes in each one of the nurse cells is stitched together to produce each one of these complexes in the actual oocyte. So we could use those estimates to set a pretty strong prior on this parameter phi, and then that essentially enabled the rest of the model to be, or the rest of the parameters to be identifiable. Okay, so then how do you go about estimating the rest of the parameters? So I said you could solve the model directly, and one of the things that you can then do is essentially look at what I would describe as like the quasi-steady distribution. So if I look at the solution to this problem, and if I look at it at long times, the only term that really dominates is this one on the far right. And then it's really this vector K2 that tells me the sort of relative distribution of mRNAs across the different cells. So essentially what I want to do is focus on using that kind of information so that I can that allows me to forget essentially about the rate constants because they're tied up. So A and B are tied up elsewhere in this model. And then that allows me to just get a handle on the bias parameter nu. And that's really, really nice, I think. And again, if you carry out the inference, what you see is that starting from essentially a uniform prior for this transport bias parameter, and sort of using the data to estimate the posterior distribution, then you can get a very good handle on the extent to which this transport is biased. And I'll say again, but a low bias parameter in this context means that everything's been on average moved towards rather than away from the oocyte. And that's what allows everything to collect there. And that's probably intuitive. And you can use a posterior predictive check just to sort of over a number of different egg chambers, just to sort of be, to check that the kind of parameters that you estimate are relatively sensible given the data you've got, and that seems to be the case. So here is everything is normalized because we're looking at this quasi-steady distribution and we're just using this vector K2. And then moving on, you can go into the full dynamic regime, and then that allows you to get a handle on the parameters A and the parameters B, which are the production rate for A and the transport rate for B. And again, you can see you can get reasonably confident estimates of those two parameters. And what you can also say from there is that in this dynamic regime, that it seems like sort of production and transport are relatively carefully balanced. So if we go back, where is it? If we go back to this original sort of system of ODEs, essentially you've got production and transport being the two terms in this equation, and A and B times the average number of Y molecules, they're on the same order of magnitude. So it's not that one of these processes is completely dominating over the other. They're both important, as I guess you might expect, but it's nice to be able to confirm that. And again, you can do a posterior predictive check where you essentially simulate the model using parameter sample from these posterior distributions, and you can compare the output of the model to the data that you have. And I've just arranged things, and it's a bit helpful later here, in terms of distance from the oocyte. So on the far right, you've got the plot that is what's going on in the oocyte, and then the next column is everything that's kind of one layer removed from that, and then the next layer is all nurse cells that are 2 away, and so on. So as you go sort of from right to left across the slide, this is nurse cells that are effectively further away from the oocyte. But we're doing a pretty good job at capturing the data, I think, with these parameter estimates. So that's all great.
So Ruth, can you just clarify? Yeah. So the points are the data.
Yeah. And
then there's a line, and then there's gray. So what's the model? Is that multiple realizations of the model, or like what we're looking at? Yeah,
sorry, I should have said. So typically, for a posterior predictive check, you're going to sample lots and lots of times from the posterior distribution and simulate the model. And then the kind of gray just indicates the range of possible outputs that come out of doing that. And then the black would be from the posterior mode, I think. and so really what you want to see is that your data is lying within the range of possible predictions that your model can make, which more or less it is. I mean, there's a few kind of outlying data points, but I think the majority of the data are, so these are the dots, the majority of the data is sitting within the sort of range of the predictions of the model. And the difference, I should say, between the red and the blue here is that sometimes Jonathan only didn't want to count what was going on in all 16 cells, so occasionally he just counted 1, 2, 3, 5, and 9, and that's why there's a few more data points in there, but nothing else special.
Okay, thanks.
That's a good point. So this is like nice, and we've got a simple model and it fits the data, and that was all great, but somehow you learn much more in mathematical biology, I think, when you get things wrong. And so what we wanted to do was to be able to challenge this model a little bit, to see how far you needed to push it before you needed to sort of refine it in order that it could sort of reproduce biological observations, because that would sort of enable us to see where our mechanistic hypotheses were too simple, and they are incredibly simple for this model. So I guess you might expect you don't have to look far. So what we did was we looked at an overexpression mutant. So effectively, if you compare wild type to the overexpressor, what you anticipate is that you're trying to have two copies of the gherkin gene in every cell. And we wanted to know if our model that we'd already sort of, we'd formulated and we calibrated to data was capable of predicting the behavior in this overexpression mutant. So the idea really is that if you've just got two copies of the gene in every cell, you're just kind of firing out new mRNA molecules at twice the rate. So all you should have to do from the model to go from the wild type model to the overexpressor model is to stick a 2 in front of the production rate, and that should if the model's correct, that should be able to capture the dynamics of what's happening in this overexpression mutant. So we collected some data from the overexpression mutant, and we used that to see whether our hypothesis was correct. So these are the model predictions. So all we've done is basically taken the parameters that we collected from the wild type, and we've stuck them into this model, and we've multiplied the production rate by 2, and then simulated the model. So this is kind of like the model predictions as context, and so the plots are kind of very much the same as what you saw a couple of slides ago. So again, this is the oocyte, and as you go from right to left, we're moving away from the oocyte, looking at different nerve cells. And the grey region shows what happens if you sample repeatedly parameters from this posterior parameter distribution and you simulate that model. Okay, and so this is the kind of this is kind of like the sort of feasible output space of that model given these calibrated parameters. So that's in grey, and red. The black line, and we'll give you what happens with the posterior mode. And so what you can see, I think, if you look closely at this figure, is if you're looking at the oocyte or one of these 4 nerve cells that are connected to the oocyte, then things are pretty good, and the model is doing a pretty good job of sort of predicting the sort of what's going on in the data. So essentially all the data points are sitting within these grey areas, which is fab. But if you go much further away from the oocyte, so to these sort of peripheral sorts of cells, so what are we looking at, 12, 16, 8, for example, then actually the model is failing pretty badly. We've got almost half the data points are sitting outside the range of mRNA counts that would be expected or predicted by the model. So things are good close by, but things are really bad far away. And so there's something about the model that now is basically incorrect or isn't working for this overexpression mutant. And the question becomes, like, what is that? One thing I wanted to say actually before I go on is that actually, although you're generating mRNAs at what you think is sort of twice the rate in these nerve cells, you're not localizing mRNAs at twice the rate, right? You're not getting double the amount of mRNAs in the actual oocyte. So, and this is just a plot that shows you over time how many mRNAs are accumulating in the oocyte. In the sort of blue is the wild type, and then red is the overexpression mutant. And to a good first approximation, it looks like they're basically accumulating. It's accumulating in about the same rate in both wild type and overexpression mutants. So there is something really quite wrong here, because again, the model would suggest that you were accumulating mRNAs at twice the rate in the overexpressor compared to the wild type. So we tried to think about what might be wrong with this. And the idea...
Ruth, can I ask a quick question? I'm sorry, that's Heiko. Hi.
Yeah.
If you go back to the previous slide, please.
Yeah.
So for the wild type, you don't have any early time points, but you do for the overexpressing. If just from your experience, if you had more wild type early time points, would the slope of that curve still be that far pointing north, or do you think it would come down?
Yeah, this is a really tricky issue, and I think a really, really good question. And we talked about this endlessly. So what you have to remember here is this is not time series data. This is data. So when you collect this data, you have to essentially fix the egg chambers. So you're sort of sacrificing and fixing. So for every dot on this represents a different egg chamber. And then what you have to do is you have to assign that particular chamber an age, right? And you normally do that or there are multiple ways to do that. And one of the ways to do that is to essentially look at, for example, the area or the volume or cross-sectional area of it, or the volume or something like that. So there's certainly issues, I think, surrounding assigning a time to each one of those. And there's to some extent, this data should be interpreted with a bit of caution, because there's a bit of horizontal leeway, I think, on every point. But it's also certainly the case that in wild type, you can't get a hold of egg chambers sort of earlier, right, at these earlier stages, and it's incredibly difficult to, for example, image them, or the processes sort of start later. So again, almost my preference would be to lose these overexpression, these early time point data, so that we're collecting data over what we think is a kind of more comparable time scale. But it's certainly a really, really good question. It would be great to have earlier time data. You also don't know what you're going to get when you do one of these experiments, right? It's like what sort of, it's what comes out is what comes out. And so yeah, it's really good question. And that's why I think I said to a first approximation, it more or less looks like it's accumulating at the same rate, but there's definitely noise in there. Maxie, did you have a question?
Yeah, I had a question as well about the previous slide. Something I was curious, there's quite a bit of variance in the sort of range of predictions you get. For example, I find for the first two distances in cells, the range is quite big, and then after that, there's some where it's very large. And for example, the top three in the middle column, there the range is very large, but that also seems to fit the data, at least kind of later in time. But then there's some where the range is very small. Is that just to do with the connectedness of these, or where does that come from?
Yeah, it's a really, really good question. I think it's possibly connectivity. I think it's possibly measurement error. I think it could be errors with staging. It's not quite clear, if you're able to shift those points horizontally slightly, that's the problem. And then the other thing goes back to the question that Sandy asked, I think quite a few slides ago, which relates to the production rate. And say more about this in a second, but I think it's almost certainly the case that when you look at this overexpression mutant, you don't have 2 copies of the gene in every cell. So it's not necessarily 100% accurate to put this 2 out here, because likely the case that you haven't got 2 copies in every cell, and there'll be some cells that don't have 2 copies. And I think that could be skewing things as well. Why that means that, well, having said that, that would probably, I guess that should probably bring these points down. So I think there's lots of different factors at play. But certainly I think all of those things combine to maybe make a difference there. If that makes sense. It's just incredibly, it is, although we've got data, it's still quite noisy for, I think, a range of reasons.
No, thank you. That makes sense.
So what was I going to say? Yeah, so I guess what we wanted to then do is go back and say, okay, if our model is not really capturing what's happening with the data, like, why is that? Because we've got robustness in the sense that it's accumulating, these oocytes are accumulating mRNAs at about the right rate, despite the fact that the nurse cells are kind of pummeling them with loads and loads of molecules. So what's kind of making that process robust? So we came up with a few different sort of ideas, and I'm going to talk about 3, but there were a number of other ones as well that we also tested and along the way. So number 1 is that there's some kind of blocking of ring canals. So simply, right, this ring canals are a pretty small kind of bridges that connect cells, and so maybe it's just that really, you know, it's like sort of a crowd leaving a stadium through these narrow sort of passageways, that there's just a build-up, there's a crowding sort of process that's really sort of, if you like, slowing down the transport process. And that's maybe consistent, you know, that's it, with the fact that you're seeing this build-up of mRNAs in kind of far away cells, because there's this blocking. It could be that there's inhomogeneous production, so each of these 16 cells has got a slightly different production rate, and that sort of overall is leading to kind of a sort of a buffering. That's very hard to measure, I should say. And then it could also be that there's density dependent transport. So essentially what happens is that as the mRNAs are produced, they're packaged up into a complex with different proteins, and they're transported on the microtubule network of the cell by essentially the sort of a motor-driven transport process. And you can imagine that, you know, it's like people catching buses. If there are too many people for the buses, then they're just not going to be able to sort of travel, right? So maybe it is this kind of density dependent transport process where the ability of the cells to transport these cargoes or complexes is just becomes saturated. So they're sort of 3 ideas, and there are others, but I think these are the ones that kind of made it through the first round of scrutiny. So the idea was that we were going to build a model for each of these processes. You can kind of have a very simple model of how they impact transport. And then use sort of model comparison approaches to think about how plausible they are. And the idea is that in terms of having a collection of models, there's kind of model 0, which is take the model from wild type, stick a 2 in front of the production rate, and off you go. And then models, that was model 0, models 1, 2, and 3 are just each of these things added to the base model. And then there's there's formal models, which are essentially different combinations of these processes all added in. So we ended up with a combination of models, or a collection of models, and we wanted to essentially use a sort of model comparison to evaluate plausibility. And we do that in terms of the ability of these models to predict unseen data. So what you do, you use the posterior predictive distribution, and essentially you're going to evaluate how good the model is by saying how well does each of these models in this collection predict data that the model hasn't yet seen. And you can use that idea to evaluate or associate weights with each one of the models, and the larger the weight, if you like, the more plausible or probable that model is as an explanation of the data. So the details are in the paper. I don't necessarily want to go through them all here, but that's the idea. So, and this is just a sort of the results. So model 0 is like the model that we investigated and shown isn't doing a great job. And then I've got a series of models with different combinations of these additional factors or mechanisms in it. And we looked at 2 different weightings. So under these BMA weightings, they're model 1, which essentially assumes this sort of blocking of ring canals. So kind of the, pedestrians trying to leave an arena type argument as being the sort of salient mechanism, just sort of this capturing the data and the processes that are sort of giving rise to this phenomenon. And that was really, really far and away the favored model. And then if you go to sort of these what's called stacking weights, then the kind of most favored model, if you like, is the one that incorporates blocking. But you can also see some support for other models. So for model 3, which is some inhomogeneity in the production rate, and for model 5, which is a combination of inhomogeneity in the production and blocking. And this inhomogeneity in production rate is just this fact that probably we haven't got 2 copies of the gene in every cell. So the mutant is not kind of quite as perfect as you might like it to be. I think it's nice. And I should say the sort of difference between these 2 ways of weighting is that the BMA weights assumed that the the correct model is in the group of models that you're considering, whereas the stacking weights sort of are a bit more conservative, and so allow for the fact that you might not have the true model in that group. And so possibly these are more appropriate because we know this is an incredibly simple model. But nonetheless, I think the real evidence is to suggest is that with the transport process, that it's blocking of the ring canals that's actually making the difference and driving the robustness of the process. And then in terms of experimental evidence, we went back. It's quite difficult to sort of get evidence for this in some senses because we don't have access to sort of dynamic data. But what we could do is go back and look at sort of what was happening very close to the ring canal. So it's hard to see where the ring canal is, but it's here in this particular egg chamber. And what you can often see is that you do get this sort of buildup of mRNA complexes very sort of close and surrounding the ring canals, which does lend some I think support to the predictions of the model, which is nice. And we haven't, I think the only thing that disappoints me now about this project is that we haven't really had the opportunity to go back in yet and sort of test this as a mechanistic hypothesis in more detail, and maybe look at some much finer sort of scale data in order to rigorously test that hypothesis. But to summarize from this bit, oh shoot, I'm also nearly out of time. Summarize from this bit, I think what I wanted to show is that although we had a very simple model, the fact that we could connect it to data using Bayesian inference made it a very powerful model in allowing us to distinguish between different hypotheses. And in particular, for this problem, I think what we're showing is that we've got a good sort of regulate or a balance, if you like, between production and transport in terms of this localization process, and that we predict that sort of crowding at the ring canals is what's helping to kind of buffer the egg chamber to kind of quite large perturbations in the production rate and allow it to be robust. So I'm going to very, very quickly, maybe in the last 5 minutes or so, switch tack and sort of try and focus a bit more on this question about like what level you want to build a model at. So in the previous kind of study, that wasn't the first model that we thought of writing down. And certainly, I guess if you'd have asked me at the start that we'd, what model we'd end up with, that would have never been the model I would say, because I would have assumed that it was effectively too simple to be able to tell us anything. But 2 points really, it wasn't too simple at the end of the day, because we could connect it to data in a quantitative way. And also that despite the fact that it was incredibly simple and that we had quite a lot of data, it was still quite difficult to parameterize that model. So we had to use some separate experimental data to essentially to estimate some of the parameters in order to make that model identifiable. So the next sort of 5 minutes, I just wanted to finish by sort of demonstrating some methods that we might think about using to develop models that are identifiable, or for which I can estimate the parameters with some degree of confidence. And so I'm going to go back to the wound healing data. So this is work that was a collaboration with Matthew Simpson at QUT and Oliver McLaren in Auckland. So in Matt's group, they have some data which looks at the scratch wound healing assays, where you don't just look at the sort of the cell population, but you also use the Fucci reporter. And what that allows you to do is label the cell cycle. So you can briefly speaking tell whether cells are in G1 phase because they look red, or in G2 SG2M phase of the cell cycle. And you can think of the sort of cell proliferation process following the scratch wound as being a series of sort of cell divisions where cells move from G1 to G2, and at the end of G2, effectively they divide to give you two G1 cells again. And so you have rate parameters Kr and Kg, which really just characterize the length of time you spend in each one of those parts of the cell cycle. And this is then what the experimental data look like. So it looks quite similar to the data that I showed right at the start of the talk, except that you don't just have kind of cells appearing gray or as one color. Now you can distinguish cells according to the portion of the cell cycle that they're in. And again, we can either kind of keep the data as being this two-dimensional data that you can see on the top row, or you can average everything in the Y direction to get these one-dimensional data profiles. And I guess they're maybe kind of, at least as a first pass, they're a nice, sort of way to go, because that allows you to build a 1D rather than a 2D model. So you've got some simplification in there. And also, I guess by averaging, you're kind of going to buffer against or decrease the noise in the signal. So what did we want to know? So in building a model of the sort of closure of this wound, or the sort of change in cell density in the scratch assay. Yeah, go on. Sorry.
So that the imaging aspect there, obviously something has to be counted. Now you can see those colors, but how are you actually extracting those distributions? Is it straightforward? Did you develop some tools for that? Is it built in?
So I guess it's relatively straightforward, because the cells are, they're not particularly overlapping, right? So it's not that difficult, I guess, to sort of count cells. And then there's a very simple, details on paper, but it's a very simple kind of averaging process where you put columns down essentially across the image, and you just count the number of cells of each type that's in each of those columns. So
how did you choose the sampling frequency though?
Sorry, go on.
How did you choose the sampling frequency?
I guess we'd used similar things before, but clearly, clearly as you change the sampling frequency, you're going to change effectively the noise and also the resolution of the data. So we didn't really play about with it, I have to say, too much, but I guess that would be a valid thing to do and a valid thing to think about. Sorry, okay. That's probably not gone.
No, it's an interesting question, I think, because it's something that, as we look at data, the scale of the data potentially is important, and we tend to measure what we can measure, right? Yeah. But it could be is that the right scale to be measuring? Should we go above that scale, coarse grain it, or should we not?
Yeah, I think it's really, yeah.
Especially talks to when we're looking at tissues where there's maybe there's this idea you're actually looking for interactions within a certain size of a domain region. And so, this idea of quadrats that comes from ecology, where you carve up the domain into a set size, how big that quadrat matters, right? How big it is. So I know this is not the same thing, but I just wondered why you chose what you chose.
Yeah, I mean, I think here we didn't think too much about it, I have to confess, but in other work we have. So often, we've looked at very similar experiments, and we've built agent-based models, and in calibrating those agent-based models to data, often, and you don't have access to that necessarily here, but often what you have is you don't just count numbers of cells and positions, but you need to summarize, if you've got 1,000 cells, it's a very high dimensional data set, so you summarize it, and we might use something like pairwise correlations in terms of distance, right, as a nice way to summarize these data, and that would tell you something about the clustering in the data, for example. And it's certainly the case that if you double the, because that's a second order summary statistic, whereas the number of cells is what I would call first order, as you double the field of view, you impact the noise in those summary statistics differently. So yeah, we have certainly thought about it. My perhaps weak answer at this point might be to say that because we're dealing with PDE models, I guess I would not be so overly concerned about it, but certainly the width of your bins is going to impact the noise, I guess, and maybe that.
No, exactly. There's a resolution below which you can't go, right? And then the question is how far above that do you want to go?
Yeah, I think so, and I think that's it's an important question, because it's sort of like, it's also maybe relative to the scale over which the data is changing as well.
No, exactly, and this looks quite sparse and clean. It might be different if they were much more tightly packed and less clean, right?
Yeah, and I think the tighter packed they are, maybe the less you want to resolve individual cells, as opposed to maybe just get a measure of density, or if it was an epithelial population, that it'd probably be quite different as well. So didn't think about it. I guess what we did do here was independently estimate the noise parameter, as I'll say in just a minute. But yeah, I think really, really important points about how you extract data from images, and essentially how you summarize those data to be able to use them in the models, I think is huge. And in the previous I guess in the previous project, we didn't really have to think about that too much because, you know, the model wasn't spatially resolved and was just counting, but here you could do more. Yeah.
Don't worry too much about the time because we always allocate an extra half an hour. So just, you know.
Okay, that's good. I'll try and be quite quick. And also at some point my kids will come home and might just wander in.
Understood.
So finishing up is good. What was I going to say? Yeah, so if you're building a model of this kind of data, then I suppose you sort of want to make use of the fact that you've got this cell cycle label to sort of differentiate between the two cell populations. But at the same time, there's a danger that when you differentiate between the cell populations, then you have a sort of additional parameters in your model, and the model is more complicated. So there's a sort of balance between kind of thinking, okay, I want to build a model that kind of sort of takes into account the fact that I've got these two cell populations that could, or subpopulations that could be sort of doing different things. And the fact that I want a simple model because I still want to be able to calibrate it to the data. So the sort of question becomes, well, if I'm going to build a model that treats these two sort of subsets, sections of the cell cycle as being cells, two subpopulations of cells, if I'm going to do that, is there enough information in the data that would be shown in these kinds of plots to merit having a model? It's going to be a PDE model, so merit having a model where essentially cells move with different speeds or have different diffusion coefficients depending on what phase of the cell cycle they're in. And I think this comes back to this question I sort of alluded to right at the start, which is like, how do I know? Now I've got quantitative data, how do I really know? Or what methods could I use to tell which model I should maybe be working with? So this goes into thinking a bit more about identifiability of model parameters. So, and you can break that down a bit. So you can say, okay, I can think about structural identifiability, which is just asking the question, if I input distinct parameter values into my model, do I get distinct distributions of observations? Okay, so that's asking that kind of question. And then in theory, it says, if I have perfect input-output data or perfectly resolved data, I should be able to determine, in theory, the values of the parameters of my model from those data. So I think that's great, and obviously that's a question that you kind of might want to ask as a sort of something underlying. But I think the problem with thinking about structural identifiability is that it doesn't take into account the fact that we don't have perfect input-output data. So the data are noisy. We can't collect a huge number of realizations of the data, and we might have sort of measurement error, etc. So I think in that context, thinking about practical identifiability is maybe more useful. The only thing is that it's sort of a slightly more woolly concept, which really just says, you know, given the data you have, can you get reasonably precise estimates of your model parameters? So we're going to focus on thinking about this model. So the idea is to have two different models, and the first model, so they're both PDE models, two populations of cells. So this is red cells and green cells as a function of X. So this is sort of distance across the wound and time. And then both cell populations have random motility modeled by diffusion, and then there's a set of reaction kinetics that represents sort of proliferation effectively. And so what's going on in here is that this term in here just represents progression from sort of G1 to G2, so at rate KR through the cell cycle. So you're losing red cells as they become green cells. And then this is the proliferation term. So in here, what's happening is that cells in G2, at the end of G2, they're going to proliferate. KG characterizes how long a cell spends in G2. And then there's a kind of volume filling term which says that a cell's only going to proliferate if there's space available. And then the factor 2 that pops up in the first equation just represents the fact that upon cell proliferation, you get 2 daughter cells. So you've got parameters KR and KG, which basically sort of characterize the length of the cell cycle in non-crowded conditions. And then you've got 2 additional parameters in this model that relate to the rates of movement of the 2 different populations. So that's model 1. And then model 2 is saying, okay, can we simplify that model? And if ineffective, or does it make sense to simplify this model to assume that cells essentially move at the same speed no matter what phase of the cell cycle they're in? And the question becomes, is it sensible to use model 1 or is it sensible to use model 2 to investigate these data? So in both cases, we're going to assume that our observation area is normal and it's zero mean and some known variance, which we can just get a handle on from the data. And that goes back to Sandy's point, I think, really about kind of how you, what bin width you use in terms of data collection. So again, going in the Bayesian framework, we've talked about already. So we want to estimate the posterior distribution of the parameters theta. So in this context, the diffusion coefficients and the rate constants, or the rate parameters, and we're going to access through the product of the likelihood and the prior. And I guess what's nice here is that if we can solve these, if we can solve these PDEs numerically, then we've got access effectively to the likelihood in this context. So the numerical bit is the solution of the PDE. Once you've got there, then you can access the likelihood. So there's sort of two approaches that you can take to doing this. The first one is that you can just directly sample the posterior distribution of these parameters using some MCMC algorithm, and then sort of not being able to identify parameters looks like very broad posterior distributions or essentially chains that don't converge. And this is the results of doing that MCMC for the first model. So the first model, different diffusion coefficients. And so the take-home message really is that for the rate constants, you get chains that settle down very quickly, and correspondingly these quite tight posterior parameter distributions. But on the other hand, for the two diffusion coefficients, then the chains really just don't appear to settle down. You can wait, as long as you want, and they're not really going to do that. So they don't settle down, and then the kind of upshot of that is you get these very, very wide posterior distributions. So you can't really say with too much confidence what these parameter values should be. And as a result, you would, I think, class that model as being sort of non-identifiable in this case. On the other hand, if you just repeat the same sort of process of parameter estimation for the second model, what you see is that not only do you get really nice estimates of both the rate constants, so the sort of cell cycle parameters, but you also get a very nice estimate in this case of the diffusion coefficient. So you get a nice sort of tight posterior, and the chains settle down relatively quickly. And in this context, this model is practically identifiable. So for me, the take-home message would be that for the data that we have from this experiment, it was sensible to use model 2 rather than model 1, because we can actually pin down and get good estimates of the parameters. And that means that we can be kind of confident of our model predictions going forward. So that's great, but it's a bit painful, right? This can take quite a long time. So for every time you're kind of trying to take a step in your Markov chain, you're essentially proposing a new parameter value. You've got to solve the PDE, and then you've got to calculate the likelihood. And it can take, there's 3 parameters or 4 parameters in these models, but this can still take a kind of relatively long time, and it's a bit overkill, maybe. So the second thing that we thought about doing was using the profile likelihood. So essentially what you want to do is you want to consider each of your model parameters in turn, and you're going to iterate over a grid of those model parameters, and you're going to look at sort of maximizing the likelihood over all the other parameters. So fix, fix the rate parameter KR over a grid of values, and for each value of KR in that grid, maximize the likelihood over the parameters KG and D, or DR and DG, depending on what you're looking at. So essentially, you're looking at the sort of curvature of the likelihood surface in that as that parameter varies. And so this sort of summarizes doing that. I think the really nice thing about thinking about the profile likelihood as opposed to sort of generating the posterior distributions or estimating the posterior distributions, that this is pretty quick. So for each value of your interest parameter, so KR was the one I gave an example of, you're doing an optimization over the other parameters in the model, and there are very well established kind of numerical methods for doing that optimization very, very quickly. So this has a kind of potential to provide a lot of information, but to do that very, very quickly. And again, not sort of non-identifiability would look like a very flat likelihood profile. So the likelihood of the parameters theta given the data is not changing. If it's very flat, it means it just doesn't change very much as you vary the parameters. So the output is not particularly sensitive to those. Okay, so let's have a look at what they look like. So this is the results of the profile likelihood analysis, and I've got everything on one slide. Sorry, it's a bit busy. But the idea is that as you iterate over, this is the grid of parameters of KR, so one of the rate parameters governing the cell cycle, then you can see how the likelihood of this parameter, and it's the profile likelihood, changes sort of as you vary that parameter. And so again, the profile likelihood is sort of nicely and quite tightly peaked, suggesting that you'd be able to estimate that parameter with a good degree of confidence because of the slope of the likelihood, or the profile likelihood. And actually, whether you're dealing with model 1, which is at the top, that's a more complicated model, or you're dealing with model 2, that's at the bottom, the same is true, right? So for both these kinetic parameters, the profile likelihood is nicely peaked. Whereas if now we think about the diffusion coefficients, in model 2, where we're assuming that both the diffusion coefficients are the same, you get a really quite nice likelihood profile, nicely tightly peaked. You'd have some makes you confident that you'd be able to identify that parameter with a reasonable degree of accuracy. Whereas if you look at the case where you've got these different diffusion coefficients, there's a real drastic change in the profile likelihood, so that it's much shallower, in particular for the green cell population, that's really, really shallow, suggesting that you won't have much, or you're not going to be able to confidently estimate those model parameters. That's just to highlight that. And so really the take-home message is that in this context, this profile likelihood analysis gives you very similar information to what you would get out of essentially using Markov chain Monte Carlo to target the full posterior, but really at a fraction of the computational cost. So this can be done very, very, very quickly compared to the MCMC analysis. So I am at the end, I think now. So take-home message really is that given the data we have here, I think model 2, which assumes that cells move at the same speed no matter what phase of the cell cycle, is practically identifiable, and then probably the one you would want to move forward with. Doesn't mean that cells don't move at different rates in different phases of the cell cycle. It just tells you that the data can't pick that apart sort of as it stands. And you might need to do other experiments to try and kind of further refine that idea. And I should say that I think both of the models appear to be structurally identifiable, so we should be able to target all of those parameters in model 1 if we had sufficient data and perfect, if you like, input-output data. So I think the take-home message of sort of both of the bits of the talk really is to think about the limitations of the data when we're building the model, so that you can calibrate the model to the data that you have. And I think identifiability analysis gives you a nice means to sort of think about sort of what level you might build your model at given the data that you have. Okay, summary I think probably is all well and good. I think I've talked over it quite a lot, and I realize I'm over time. So I just want to finish by thanking the people who did much of this work. So the mRNA localization stuff was all done by Jonathan Harrison when he was a PhD student in my group, and he's now a postdoc in Warwick. And it was in collaboration with Elan Davis and Richard Parton, who's in Elan's lab. And then the last bit was all in collaboration with Matt Simpson and Oliver McLaren in Auckland. So thanks for your time, and I'm happy to answer any more questions if that would be useful.
Excellent talk, Ruth. This was amazing, and obviously you have had a few questions already. So I think that some of the most urgent things have already been sort of asked, but I'm pretty sure that there's more people who have maybe following up questions.
I think so, yeah.
I think Jan has a question.
Yeah.
Yes, I do. Thank you. Thank you, Ruth, for a great talk. I really enjoyed it.
Thank you.
I missed such an excellent presentation. So I've got a question to the first model. So assuming the structure of the graph, there is you've got like 4 sources for the not the nursing cell but the oocyte, right? So you've got 4 sources, but they have different sources themselves. So basically, if you look at the graph, right? So from the cell number 5, so cell number 5 is fed only by the cell number 13, right? And cell number 2 is fed by a lot more cells, right? So basically, if you assume that in each cell you've got the same production rate, right? And then the bias towards the oocyte is the same, then you should expect more inflow from the cell number 2 than from the other cells, right? So my question is, you've got the Z-stacks from the microscope. Do you see that there is a large amount of mRNAs closer to the boundary with cell number 2? Because there should be more mRNAs coming from them compared to the others, and that could be additional validation about your assumption of equal production and equal bias, right?
Yeah, so we didn't look at resolving within a nurse cell kind of where the mRNAs were, but I think you can see it. So the dots here are actually the data, and so you can see that actually 2 is on quite a different scale from 3 and 5 and 9, right? So 9 is only being fed by one other cell, and actually the scale is a lot lower. Whereas I think 5 and, get this right, 5 is being fed by more than 9, and then 3 by more, and then 2 is the one that's being fed by loads and loads, and the scale is much higher. So I think that does come out in the data, but I think I think it's a really nice way to think about it. I think it's captured anyway, in essence, by the fact that and you can see it. You should be able to see it in some of these density profiles. It's quite hard to see, but it is there, and I think that is naturally captured by the model and in the solution to the model. And actually, it comes out in K2. So essentially, everything else over here is like kind of initial condition related to the initial conditions, and then this is really the parameter this is really the term that captures what happens in what I would describe as the quasi-steady distribution. And at that point, it's the distribution of the distribution is governed by the vector K2, which will encode all that information. Does that make sense?
Yes, it does.
Yeah, I think that's what allowed us to kind of get a handle on the bias parameter so well, was that we effectively, here it is, we looked across all the egg chambers that we had and normalized everything to the amount that was accumulated in the oocytes, and that allowed us to get a handle on K2, and therefore knew and throw away all of the rest of the information, which was quite useful.
Sure, thank you.
Cool. I think Johan's next.
Ruth, that was a dynamite seminar. Thank you so much. I actually really enjoyed all of the elements. So if I can take a question to a slightly higher level, sort of philosophy is, a number of scientists have pointed out that when you're building models, you're often going for precision, generality, and realism. And you can have 2 of them, but never quite all 3 of them. And in watching what you're doing here, it seems like you're creating a recipe for getting strong precision, but have you sacrificed realism, i.e. the why of this?
Probably to some extent. So I think what's interesting to me is that in both of these examples, like quite a slight tweak to the model, or even in this context, building an incredibly simple model, it was still quite hard to pin down the different bits of that model given the data, and we had to work quite hard. So that also says to me, okay, let's, I'm a real fan of, like I said, I think in the talk, I would never have thought to start this simple, but I think starting this simple was still valuable. And I think there are other models that you can build on top of this that kind of interrogate more detailed questions. So there are models of what happens in terms of transport in a single nurse cell, and we develop models as well of what happens in terms of transport in a single nurse cell. But I guess I don't think they're models that would answer, I think, different questions. So for sure, we've sacrificed a lot of what we know about the biology to make the model simpler. But that didn't mean that we couldn't get insight into what was happening. And I think we sort of came to the realization that any more complicated a model would not allow us to kind of calibrate it to the data. And in this context, it was really that calibration process that helped. And we've got kind of other projects that are on the go at the moment where we have as well tried to do these model comparison processes. And I think sort of the calibration is really important, and being able to do kind of model comparison. I think if in this context, if we couldn't have pinned down the model parameters, we wouldn't have been able to tease apart those three different mechanisms in terms of their ability to describe the data. So yeah, I totally agree. And it doesn't mean that doesn't mean I'm against building much more complicated models, but often I think those comparisons are qualitative, or might be, what's the word? You can use to explore a range of potential mechanisms.
Right. So Ruth, I guess part of the invitation or thought or response to my question actually had less to do with building more complicated models as to using different model premises. So ordinarily in cellular function life, the movement of, say, a trail of ants, if you're watching a trail of ants on your countertop, you can do scratch assays, and people have to see how does the ant kind of line of ants get messed up and re-equilibrate and whatnot. And one could build a PDA model, and one could model the dynamics of what the ants are doing and all of that. However, another approach to the ant colony, this trail of ants, is to actually structure the model as an economic model. Why are the ants in a line like this? And then you'd understand, oh, they're going to the sugar bowl. And why does your scratch assay muck things up? Well, they're afraid of predators and individuals being lost. And so I think it's less, I guess my query here was less about making the model more complicated in terms of the existing ones, but rather fundamentally deciding what is the attribute and processes you're trying to understand. And so the alternative to all of this, if it's a life model, is there's usually some sort of cost-benefit decision being made.
Yeah, for sure. I mean, yeah, we certainly didn't frame it in that context, and I guess maybe one could think about what that is, and so maybe you might have a criterion that tries to think about kind of ability to buffer against insults or robustness, or maximizing accumulation rates. Right. Yeah, I'm not sure. I guess, yeah, I think it would be, yeah, I think it's a really interesting idea, and one we're thinking about in terms of how you frame it. So yeah, great question.
Well, Ruth, thank you. Thank you for entertaining the discussion on this.
Thank you.
Audrey?
Hey, this was a great talk. Thank you. I'm wondering, I have a question about sort of the biological relevance of the wound healing assay. I'm just not very familiar with them in general, but it seems like, so at least for your first model, I'm thinking about slide 36 in particular. Thanks.
Yeah, yeah.
So at least for your first model, so I kind of was noticing the rays on the edges too of the models of the cells in different phases, which I'm guessing, is that correlated to cell growth?
So where are you looking along the edge of the wound?
Yeah, so particularly in the bottom at the quantification.
Yeah.
So those rays along with the area that's filling in. So near X of 0 and X of 1242. Those, those values are raising over time, it appears.
Yeah.
So is that a side effect of cell growth in the assay itself?
So I think it can be multiple things. So traditionally here we'd use a zero flux boundary condition at the edge of the assay, and we do that whether it's a PD model, I think, or an agent-based model. But some things that we have done in the past are to do things like, kind of double the extent of the model domain and just check whether that boundary condition kind of has any impact on the solution. So I think in here it's definitely the case. I think that if your population is not quite a confluence when you start the experiment, you're going to see proliferation kind of route at the edges, as well as kind of you sort of, I think you traditionally think it's all going to happen in this area here because there's much more space, but I think unless you start at confluence, there's still room for proliferation behind the front as well, and I think that's what's going on certainly here. And in a way, I think if there weren't otherwise you would get like a depletion of cells behind, right? So if they're moving quicker than they're proliferating, it would sort of tend to deplete at the edges in a way, because as cells moved in to fill the available space, and I think that tells you that proliferation is overcoming that in this context.
Okay, so this isn't this sort of edge effect isn't believed to be biologically. So if I did this sort of assay on a mouse, right, I wouldn't see this sort of edge. Okay.
No, I mean, I think, I guess going back to these pictures, this is kind of precisely what, it's kind of funny whenever we're working on another project at the moment where we've got loads and loads of these data sets, and the dish is huge compared to the field of view that you actually use in the experiments. But I guess what you are kind of confident about is that there aren't necessarily edge effects from the sort of dish or anything like this. So this is representative of what's going on. But I think it is important to sort of check that the boundary conditions you impose on the model aren't having because essentially when you build your model, it's just of what you can see in the image, right? And so you need to be a bit careful that you're not imposing boundary conditions on the model that are impacting upon the model predictions, because in the dish it's different, and the field of view is and the assay is much bigger than that. If that makes sense.
Okay. Oh, thank you.
Great questions.
Cool. Well, thank you very much for your question.
I have a quick question.
Okay.
Hello, I'm Daniel. I'm in Heiko Enderling's lab, and my question with the practical identifiability with the Bayesian inference, how do you determine? Is it just by eye that you determine? Okay, the posterior is flat enough to say that it's not identifiable. Is it just by eye, from experience, that kind of thing?
So you can essentially threshold it, right? Which is I don't know if we mark this on here, but you can look at you can more formally quantify when you would characterize something as being identifiable or not by putting essentially sort of confidence intervals down over this, right? And then what you need is that your what am I trying to say? So that would give you some kind of quantitative measure of sort of identifiability in that context, but it's still then a matter of choosing like that threshold level, right? And there's accepted sort of choices you can make there, but for sure, I think in terms of practical, you can quantify it, but it's always slightly subjective in terms of what thresholds you pick to accept or not. I think for me, I mean, it's still a very useful concept in terms of thinking about the range of parameters that would be useful to consider. And maybe I think often actually these kind of identifiability analyses can tell you maybe about ways to simplify your model, or ways in which you might want to simplify your model given the data you have. So you couldn't kind of do. So I showed only kind of univariate profiles, but you could do a bivariate profile where you looked at a grid of 2 parameter values, and then what you might see is that there's a ridge in kind of profile space, and that would basically tell you that you've got 2 parameters that sort of trade off against one another, and you might want the product or the quotient of those parameters might be a way to kind of reparameterize your model to make it identifiable, if you like. So I think there's loads of things that kind of doing this sort of profiling can tell you about how to simplify your model to make it identifiable. But it doesn't mean it's the model that you have to sort of stick with, because I think one of the nice things about it is sort of saying, well, okay, at the moment it's not identifiable, but what if I did this experiment and collected this data, or these data, would that make these parameters identifiable? And if it does, then you can test that kind of in silico, and then you can go back and say, okay, I think this is the data we need to collect. Or you might, ideally you'd do it before the fact, and you would say, okay, I need data at this time or this spatial resolution, temporal or spatial resolution, in order to be able to make the parameters of my model identifiable. So I'd sort of think of it as a tool to think about what level to pitch the model, but also as a tool to think about what experiments are quite useful to do to pin down the parameters. That makes sense.
All right, thank you. Yes.
So I have a question, unless you have one, David.
No, I'm fine. Good.
Okay, Ruth, thanks. That was great. It was great to hear some of your research in person. And I know we interact a lot because of your role in the board of directors of the society, which is really important. But we don't really get a chance to talk science. It's been nice to see some of your science, and it's very cool. This part, second part, focused on the parameter identifiability. I think, you know, we wrestle with this a lot, and it's partly philosophical, but it's also partly practical. And I think Joel's point sort of hinted on that. But maybe where it becomes very useful is when you do have a data set that's quite, let's say, accurate and well derived, and you want to really say dismiss certain hypotheses or say that this is not true, but this is true. And how confident could you be with that? And I think this sort of, as you say, identifiability potentially gives you to say, well, you can't make an assumption about this because you can't actually pin down the parameter. Right?
Yeah.
Even if the fit still looks great, so to speak, right?
Yeah.
But we also use models a lot for hypothesis generation, and often in scant data, right?
Yeah.
And there's no way to use these tools then, right? So there's a time and a place to use them for sure. I think it's important, and we need to develop this sort of tool set and build on it. And it's something that as a field we need to think hard about, right? Because it does push against the, I think I know what the biological processes are, and so I'm going to add them in, versus I'm going to ignore all the biological processes and just get a really simple model that I can clearly parameterize, right? And so there's a push and pull there, right?
Yeah, I mean, I think it's, one thing to highlight is to be by no means within my group is this kind of the only way we do things, and there's certainly projects that are much more kind of exploratory and focused on hypothesis generation and testing and really kind of explorations, and I think some of that comes down to kind of how much like prior knowledge or how much existing knowledge you have about a system. So we have a project, one relating to kind of drug toxicity, so totally different to this, and another one relating to sort of cell competition and the mechanical aspects of that, where I think in both those cases we know like far less about well, certainly with the cell competition one, we know far less about the actual kind of mechanisms, and it's much more about developing a model and encoding lots of different ways in which the cells can compete with one another into that model to then think about the kind of range of outputs that we could see. And so that's really about hypothesis generation. But in this context, I think for some of the examples I showed, because we know a bit more and the data is much more well resolved, that we're in a position to sort of develop models in slightly different ways and to use them to answer slightly different questions. And so I think it's incredibly important to be to recognize that both are incredibly useful tools, and it depends on the question, it depends on how much you know, and it depends on kind of how much data you have as well, I think, as to where you go. And hopefully at some stage we'll begin to join the dots on more projects, but I think there'll always be a place for sort of both types, right, of approaches, very much so, and that's a good point.
Really support that. We need both. But I also think, as you say, maybe there could be some future convergence, and that could actually be where in a system where you can sort of do this type of analysis and you actually pinpoint there's an area that you can't separate something, a process, that should drive different experiments that might facilitate that separation, right, and allow a more deeper understanding. So there could be some nice feedback there too.
Yeah, I think precisely, and for a long time I worked kind of solely with sort of qualitative models and qualitative comparisons, and you're still able to, in those contexts, make predictions and to make suggest experiments that would help you kind of further refine your knowledge and understanding, and you can do the same in that context as I think you can do here. It's just using different tools to do that, really. And I think importantly going forward, thinking about bringing all of those different aspects together, and like you say, focusing on hypothesis generation in areas where you're more unsure about aspects of the model is really important. And the other one I think is about thinking about how to, in more complicated cases, learn models directly from the data as well, which is kind of another step. across.
Some of the other work that you're doing is very good.
Yeah, so we're certainly thinking about that a bit as well, which is maybe more kind of non-traditional in terms of this idea of there being, you know, an art to sort of model development, which I don't really think there is, but we're sort of taking that, you know, yeah, taking it straight from the data.
Yeah, I just want to chime in and say I truly appreciate the effort to develop simple models and to do a proper analysis and what's identifiable and what is not identifiable, and you know, what can you drop from a model that doesn't inform or contribute any information given the data that you have. So it was very beautiful to see the Bayesian approach to these kind of questions that we just started dabbling in. I don't know if Stefan's on the line, who's trying to do something. So we look forward to maybe chatting a bit more in the future.
Yeah, that would be really great. Yeah, yeah. Fantastic.
Adding to all the other Zoom meetings that we're having already that have nothing to do with science. Thanks anyway.
Thank you.
Great. Well, David, I think we're good to wrap up.
Yes. Thanks, Ruth, once again, and thanks for this very thought-provoking presentation.
Thanks for the invitation, and thanks for everyone for all the really insightful questions and comments. It's really valuable. Thank you.
Bye.
We look forward to having you here in person in the future.
That would be great. Yeah, in the future. Take care, everyone. Whoa.
Okay. Thanks.
Bye.
Bye.
Thank you so much.